Hello, good morning, everybody. Welcome to the morning session of YouTube live stream. I'm Mrs. Priya Mohorkar from SV Union High School and Junior College, Pune, Maharashtra, India. Today we are going to continue with Standard 10 Science and Technology Part 2, Chapter number 8, Cell Biology and Biotechnology. Let us start with whatever we learned in the last session. Okay, let us just recall certain things. Last session we learned about that all the living organisms are made up of cells, and we also saw that the hierarchy of organization of an organism, that is cell, all the organisms are made up of basic structural and functional unit called as the cell, right? Whether it is unicellular organism or multicellular organism, the basic building blocks are the cells. When the similar cells group together and perform the same function, they form what are called as the tissues, right? These tissues are grouped together. Different types of tissues are there in plants as well as in animals. The basic types are in plants are the meristematic and the permanent types of tissues, whereas the basic types of tissues in animals are 4 different types, as you have learned in the lower classes: the epithelial tissues, the nervous tissue, muscular tissue, and the connective tissues, right? Of which epithelial is the simple, and all the other 3 types are the complex types of tissues, right? When these different types of tissues group together, they form what are called as the organs, right? Different types of organs, like you have your stomach, the liver, brain, heart, right? Intestines. You have different types of organs that may be grouped together. Now, certain organs group together and they form what is called as the organ system, right? Organ system you have learnt about the few organ systems in man, that is the digestive system, which is the basic and the first organ system that we learn in the lower classes. So different organs together form the organ system, whereas such different organ systems like the digestive system, the nervous system, the respiratory system, the circulatory system, right? All these different types of organ systems are going to form what are called as the organism as a whole, right? So the hierarchy of organization of an organism we have seen in generally in animals, but in plants also it is going to be the same. The plants are also made up of cells, tissues. Different types of tissues form different types of organs, like the roots, the stem, the leaves, the branches, the flowers, the fruits, and the seeds, right? So vegetative organs as well as reproductive organs. These organs together would form the organ system, that is the root system and the shoot system. Whereas the root system and the shoot system together form the whole organism, which is the plant. So whether it is going to be in plants or it is going to be animals or human beings, of course human beings are social animals, right? The hierarchy of organization is in a similar way, right? Is similar to all these complex organisms, right? We talked about biotechnology now, which is a new field of science, which is a branch of science in which the techniques of bringing about improvements in living organisms by artificial genetic changes and by hybridization for the welfare of human beings, together called as biotechnology. Now, why suddenly this biotechnology we are talking about? Because we can make changes, bring about changes in the basic structure of a cell of an organism. When there are certain changes, certain modifications that are being brought about in the cells, in the genes, in the DNA of the cells, then it is going to make changes, bring out changes, and of course the expression would be the desired characters, which we are learning in this field called as the biotechnology. In today's world, biotechnology plays a very important role in every field, mostly the agricultural field, the farming, the animal husbandry. Not only that, even the microbiological industry, the pharmaceutical industry, the health industry is all dependent mostly on biotechnology. Why? Even now the vaccines which are being produced for the against the COVID-19 pandemic is also the product of biotechnology, right? So let us see what are the different techniques under this under this new field of science, that is biotechnology. Now, biotechnology actually has two basic methods we learned in the last class. First one is the tissue culture, which is a basic method. Second is the genetic engineering. Of course, genetic engineering still has few more methods in it, under it, but these two are the basic methods which can be utilized, and the techniques can bring about improvements in the living organisms by artificial genetic changes. Okay, let us see what did we learn in the tissue culture. Now, tissue culture can be done both in plants as well as animals. This process is actually an ex vivo growth of cells, that is, externally the living systems are grown in aseptic conditions in nutrient rich medium, okay, and these cells are grown in controlled conditions, that is in the lab, right? So this process of ex vivo growth of cells or tissues in an aseptic and nutrient rich medium in controlled conditions is called as tissue culture. Now, this tissue culture process was possible and is possible due to the totipotent or the pluripotent capacity of the plant or the animal cells, right? Now, what is this totipotent and pluripotent capacity? Now, totipotent capacity is the total potential, total capacity to develop into a new whole organism. Now, this capacity is present in the cells. Of course, with the help of this tissue culture technique, a complete organism can be developed from a single cell or a group of cells, which we call it as tissue. So, from a single cell or a tissue, we can grow a whole organism. That is the power of life itself, right? So we saw that if at all explants are taken from the leaf part, vegetative part of a plant, they can be grown in the nutrient medium that grows and multiplies. The cells grows and multiplies in the nutrient medium and form what is called as callus. Callus is a group of irregularly growing cells, fast growing cells, which are undifferentiated cells, right? The differentiation will be later on provided by the growth hormones, and of course the shoot multiplication of these callus cells into the whole plant, into a small plantlets, and then later on into a whole plant is possible in the tissue, in the culture medium. Now, this plantlet undergoes another process, hardening process, and after primary hardening into the pots, small pots, which are grown in so that the plantlets can be grown in the soil, and of course the later on under secondary hardening treatment, they are slowly acclimatized into the natural environment. Right, under this tissue culture technique, we talked about the stem cells research, right? After cloning, actually it was a revolutionary event, and this technique actually brought about fundamental changes in the field of medical sciences, right? Now these stem cells are basically of two different types. One is embryonic stem cells, the other one adult stem cells we learned. The stem cells are generally used in the formation of various organs like the brain, the liver, the heart, the bones, right? Or different types of cells like the hepatocytes, the neuron cells, the bone cells, the cardiac cells in such manner. Other uses of stem cells is the regenerative therapy, under which we saw about the cell therapy, in which the stem cells are used to replace the dead cells in case of conditions like myocardial infarction, diabetes, Alzheimer's disease, Parkinson's disease, etc. Not only that, they are also used to produce cells like anemia, thalassemia, leukemia, etc. Right? So you can see here the picture. I explained the picture of the myocardial infarction, which was recovered by regenerative therapy. Other uses of stem cells is formation. When the organs are regenerated, they can be transplanted. So under organ transplantation, in case of any failure of any organ like kidney, liver, they can be produced with the help of stem cells. Like here we have a small picture showing the regenerated ear pinna, and that was transplanted successfully in recent time. Let us talk about genetic engineering now. The deliberate modification of characteristics of an organism by manipulating its genetic material, which is the DNA, right, takes place, is done in this process, in this method of biotechnology called genetic engineering. You can see the genes are manipulated, maybe added, deleted, or maybe their position might be changed so that new organisms may be formed. Not only organisms, new products might be formed. Let us see, today let us just have a look at the general process of genetic engineering, right? So the general process of genetic engineering, if you see in plants, how it is being done. Now, generally, small parts of the maybe vegetative, embryonic, or even the reproductive part, the buds, okay, such kind of plant parts can be taken, and the explants are being transferred in the nutrient medium after the process of sterilization in aseptic conditions. Now, once in the nutrient agar medium, the explant starts multiplying, right, and form a callus. But during that process, the microorganisms play a very important role here in the process of genetic engineering. Now, what is that role played by the microbes? Now, microorganisms like the bacteria, generally Escherichia coli or the agrobacterium, such bacterium are used as experimental microorganisms. Now, in which the desired gene can be incorporated, right? And these bacterium can be introduced or allowed to infect or introduce the gene into the explants which are growing in the nutrient medium. Now, these explants now which are forming the callus, but along with the bacterial genome or the gene, the desired gene which get incorporated into the explants. Now, such callus that is formed will be called as transgenic. Okay, the cells of this callus will be called as transgenic cells, and these transgenic cells can be later on grown into small plantlets by the addition of the growth stimuli or the stimuli providing hormones into the roots as well as into the shoots. Okay, now these plantlets that are grown are called as transgenic plantlets that can be later on put into the hardening process, both the primary hardening and the secondary hardening. Now, this plant which survives the hardening procedure is going to be called as a transgenic plant, in which the desired gene is is found into every part, every cell of the plant. So this plant will be called as the transgenic plants. You have learnt about the transgenic plants, heard about at least transgenic plants in the lower classes. So this is how the general process of genetic engineering takes place in the case of producing transgenic plants. Now these can also be called as the genetically modified plants. Generally, crop plants are being genetically modified for better yield and to withstand the stress, different types of stress like temperature, drought, okay, any kind of disease resistant, all these kind of stress can be withstand by the new transgenic plants or the genetically modified plants. So under this field, the biotechnology, there are various branches of science that are included. Why? Because we need to study the basic structure and function of every type of cell. Under cytology, biochemistry, under biochemistry we need to learn the different types of biomolecules present in the cell or produced and synthesized by the cells, or otherwise the byproducts that are being produced under different conditions by the cell. So during the regular cellular processes. So this can be studied, and this can be studied under biochemistry, which is also necessary for biotechnological processes. Not only that, molecular biology and genetic engineering should also be studied, and they are directly related to the biotechnological processes. All these branches of science are related to biotechnology. I would say included in biotechnology, because the study of these structure and the functions of the cells, the products, the byproducts produced or synthesized by the cells are very, very essential. The basic physiological processes of the cells is very, very essential to understand and implement the biotechnological processes to those cells. Now that is why the various branches of science are related to biotechnology. Now they are also included too in biotechnology. There is a considerable progress in the field of agriculture and pharmacy due to biotechnology. New experiments are being performed for improving agriculture yield. Of course, in the field of agriculture, lots of experiments have been done, and the scientists were successful in producing many genetically modified crops, high yielding varieties crops, hybrid seeds, plants. Okay, so a lot of work has been done on this. Now, whereas in pharmacy and medical sciences, experiments for the production of antibodies, vitamins, vaccines, hormones, all these biomolecules are being studied on. Experiments are done, and these biomolecules are being produced. Hormones like insulin have been successful, and nowadays you get insulin in large quantities because of the biotechnological processes, so that they can be used in health sciences, mainly for diabetes. And even you know now that vaccines are being produced by the biotechnological processes for even COVID-19 pandemic against the coronavirus group of viruses, which are the coronavirus which is causing this pandemic. Anyway, high-yielding variety crops have also been developed by the method of tissue culture techniques and under biotechnological field. So let us see which are the main areas included in biotechnology. So biotechnology includes the following main areas: use of various abilities of microbes like yogurt production from milk and alcohol from molasses, sugar molasses, of course. Now, so microorganisms are being used, their capabilities are being used widely, of course, in the production of milk and milk products. Not only that, even production of alcohols from sugar molasses. Not only that, the use of productivity of cells is also the major area of biotechnology. For example, production of antibiotics, production of vaccines, etc., with the help of specific cells. Now, use of biomolecules like the DNA and proteins in human welfare is also a main area where biotechnology plays a very important role. Development of plants, animals, and products of desired quality by gene manipulation that is genetic engineering, is an also very important area under which biotechnology plays a very important role. Production of human growth hormones with the help of genetically modified bacteria. That is a different technique wherein the genetically modified bacteria helps in production of growth hormones, not only growth hormones, other hormones also, like insulin. The use of genetic as well as non-genetic techniques or also fall in the main areas of biotechnology. Non-genetic biotechnology actually involves the use of either cell or tissues. For example, the tissue culture, production of hybrid seeds, the use of stem cells in the production of organs. All these things are important areas of biotechnology where biotechnology is generally used. Let us see the benefits of biotechnology now. Benefits of biotechnology, of course, for the welfare of mankind. It has become possible to increase the per hectare yield irrespective of the limitations of cropland area in the field of agriculture, of course, the simple tissue culture technique helps in multiplication of the crop plants. So the limitations of course are there under the cultivated land or under the agricultural land area, but now because of biotechnology we have increased the per hectare yield, and of course the consumption of food is almost to a greater level nowadays. Expenses of the disease control have minimized. So disease control expenditure is minimized because of the production of development of disease resistant varieties of crops, so that the crops, when disease resistant varieties of crops are being developed, the yield of crops has increased, the production of food has increased because of this. Otherwise, initially, due to various diseases, fungal, bacterial, or viral diseases, the crop plants would get damaged. And of course, that used to affect the production of food, as well as consumption used to be was also affected. Now, due to the development of fast fruit setting varieties, now fast fruit setting varieties are other group of varieties where the setting of fruit, that is flowering and fruit setting, is faster, takes place early than the natural normal period of development of the plant or the crop plants. The yield per annum has also been increased for the fruits or the seeds, whichever the plant yields, and the development of stress resistant varieties which can withstand the variable temperature, water stress, changing fertility of soil, etc., has become possible. Now, in India, let us see how this field of technology has developed under this heading, development of biotechnology in India. Government of India has established the National Biotechnology Board in 1982 that was transformed into Department of Biotechnology under the Ministry of Science and Technology in 1986. Now, this department includes in certain institutes like the National Institute of Immunology, National Facility for Animal Tissue and Cell Culture, National Center for Cell Science, National Brain Research Center, Central Institute for Medicinal and Aromatic Plants, etc. Now, let us talk about the commercial applications of biotechnology. Now, I said we are um, surrounded by biotechnological products, and we most probably consume biotechnological products nowadays. So let us see what are the various commercial applications of this field, biotechnology. Actually, biotechnology plays a very important role in different fields. Let us see. They are the crop biotechnology, the animal husbandry, human health, industrial products, which is termed as the white biotechnology, environment and biotechnology, that is the role of environment in the role of biotechnology in environment, the food biotechnology, and the DNA fingerprinting. Let us start with the crop biotechnology. The biotechnology used in the field of agriculture to improve the crop yield and crop varieties comes under crop biotechnology. The process of genetic engineering is basically used in this under crop biotechnology. What exactly is done is genes are modified that results in the production of desired characteristics in the crops. So crops are genetically modified, of course, by various trial and error methods. This has been done, and once the successful expression of the modified genes, the genetically modified genes takes place, then the desired characters, and as the desired characters are expressed by the crop plants, the genetically modified crop plants are being developed, and they are being grown and of course consumed. Now, the next area is the hybrid and the tolerant varieties, which are developed by few techniques like hybrid seeds, genetically modified crops we have, and the biofertilizers. Let us talk about the hybrid seeds first. Actually, the hybrid seeds are being produced by the recombination or combination of genes of different crops, okay, so that the desired characteristics of those crops can be attained in a single crop. So the seeds which are being produced after the recombination of the genes of two different crops for desired characteristics, these hybrid seeds are being collected and they are being grown for the hybrid genetically modified crops. Especially this process is used in the and is applied to the fruit plants, okay, fruits and seeds plants. For example, you can see nowadays the purple watermelon is in, of course, in the market. Everybody is astonished actually to see purple watermelon. You have yellow watermelon, you even have blue watermelons, if you think of just watermelon, just one variety. So not only that, the seeds of these plants can be collected and can be stored in the seed banks. And of course, you have different hybrid varieties of seeds that are being produced nowadays in case of fruit plants, okay. Not only fruit plants, even in the case of pulses and cereals. Right, so let us talk about the genetically modified crops here. Crops which are developed with desired characters by integrating a foreign gene with the genome of these plants is called as the genetically modified crops, right? The high yielding varieties with resistance to diseases, alkalinity, weeds, stress resistant like cold resistant and drought resistant varieties are grown. For example, you have genetically modified BT cotton. Biotechnology, it is Bacillus thuringiensis cotton, BT cotton it is called as generally. The golden rice, which is in the market nowadays, which is said to have the largest amount, the more amount of beta carotene, and the herbicide tolerant plants, etc. Now all these are the genetically modified crops. In the lower classes, in class 9 you must have, or ninth grade, you must have learnt about more about these genetically modified crops. Next, let us talk about this BT cotton and BT brinjal. Now BT cotton, what is this? It is actually a genetically modified crop, cotton crop, in which the gene, a particular gene from a bacteria called as Bacillus thuringiensis. Okay, the name of the bacterium is Bacillus thuringiensis. Now this gene was integrated with the gene of cotton, and as well as, of course, brinjal later on. Now this gene actually produces a toxin, a poison toxin, a chemical substance. It produces a product, okay, a protein. Now this protein actually is toxic substance to the bollworm. The bollworm actually feeds on the leaves of this cotton, general wild cotton. So this cotton species used to be damaged by the bollworms, right? The pests. So this particular cotton, when it was introduced, or when it was integrated with that particular gene, we simply called as BT gene, starts producing the toxin, that chemical substance, which was actually produced in the cells of the leaves. Okay, and of course the bollworm feeds on the leaves of the cotton. Now when this particular gene produces the toxin, the bollworm, if at all it feeds on such leaves producing the toxin, the bollworm died because of the damage caused to its alimentary canal. Now that is how they started using this particular BT gene to incorporate in other crop plants also, like BT brinjal. And of course they were successful, scientists were successful in producing not only BT cotton but also BT brinjal. So if the bollworm tries to feed on this genetically modified crop leaves, then the toxin that was produced would destroy the bollworm, of course. So the plant was itself resistant to this particular pest, which used to damage the whole crop, called the bollworm. Okay, so the BT cotton surely was resistant to bollworm once it was genetically modified by that particular gene from Bacillus thuringiensis bacteria. Okay, so let us talk about another product of biotechnology here, that is the golden rice. Golden rice, actually the gene that synthesizes vitamin A, the beta carotene, has been introduced in rice. Okay, now of course the gene is being introduced into the rice, and the rice slowly, when it grew with this particular gene in it, started synthesizing vitamin A 23 times more than the normal variety of rice. So here you can see the difference between the plain white rice and the golden rice. Different types of rice. You can see the wild type rice, which is white in color. Should I say colorless, or no, whitish, dull white, or say off white in color. But the golden rice type 1 and type 2 here shows the difference of color. Now that is because of the beta carotene that is produced in the rice grains, right, after being genetically modified. So golden rice, are you having it? Yes, start having a golden rice, at least a small amount, so that you can get vitamin A from the rice itself. And rice being staple food of most of the Asian countries, of course, would benefit human beings, isn't it? Next is the herbicide tolerant plants. Now, herbicide tolerant plants were also developed by biotechnological process. Um, why and what made people or scientists produce such type of plants? Now, generally, along with the main crop that were grown in the fields, the problem was of the weeds which were grown, which were unnecessary plants. Weeds are actually unnecessary plants that grows along with the crop. Okay, so these weeds affect the growth of the main crop. Now, how do they affect? The weeds are very fast growing plants, okay, and they always show lots of vigor in growth. So, does not allow the main crop plants to grow near them. And there are, of course, many other factors, physical as well as chemical factors, environmental factors also. But these weeds always affect the growth of the main crop. Now, when suppose herbicides are used, so herbicides to kill the weeds, okay, so that the weeds can be destroyed and can be removed from the crop fields. But the herbicide that are used may also affect the main crop. So in that case, herbicide resistant varieties of crops were developed by biotechnological process. And these herbicide resistant crops actually selectively destroy the weeds. Okay, so herbicide tolerant plants were also developed using biotechnological techniques. Now, the next field where crop biotechnology is useful is the biofertilizers, the use of biofertilizers. You all know bio refers to bios, refers to living organisms, mainly the microorganisms. Okay, not only microorganisms, it's all the living organisms, whether plant, animals, human beings, microbes, whichever organisms are living. Bios refers to living organisms. And here, fertilizers are those here living organisms are those living organisms which actually help in enriching the soil or enriching the plants with more and more nutrients. So such organisms are called as, such living organisms are called as biofertilizers. Okay, now due to the use of biofertilizers instead of chemical fertilizers, generally chemical fertilizers were being utilized. Should I say were being utilized? Even now they are being utilized. Chemical fertilizers are utilized for better growth of crop plants. Okay, like the urea or mainly rich in nitrogen, phosphorus, and potassium. So such fertilizers, the chemical fertilizers, are utilized for better growth of the plant, for better yield of grains or of the crop. But these chemical fertilizers slowly started polluting the polluting the soil by making the soil more saline, more acidic, right? Because of this retainment of the chemical substances which were used as chemical fertilizers. So instead of good growth and good yield, the crop plants started deteriorating in their growth, and the yield also started reducing. So nowadays, instead of chemical fertilizers, biofertilizers are being utilized, mainly in the organic farming. Of course, biofertilizers are used. Now, which are these biofertilizers? The biofertilizers are those which help in fixing nitrogen, that is nitrogen fixation, and phosphate fixation ability of the plants, that are being improved by these biofertilizers. Mainly bacteria, the microorganisms like bacteria and cyanobacteria are being utilized in providing the nitrogen fertilizers to the plants, to the crop plants, through bacteria like rhizobium, azotobacter. You must have heard these names of bacteria. Not only bacteria, but there are other group called as the cyanobacteria, the blue-green algae, the filamentous blue-green algae, which are which generally help in the in the fixation of nitrogen into nitrogenous substances for the plants. Uh, Either in symbiotic relationship or otherwise being free living. Also, they fix nitrogen, atmospheric nitrogen, into nitrogenous compounds and let them into the soil so that the plants growing in that soil can be benefited. Now, these microorganisms generally act as the biofertilizers and are called as biofertilizers only. So not only microorganisms, some certain plants like azolla, which is a pteridophyte, which is a plant that actually is in symbiotic relationship with the cyanobacteria, namely Anabaena azolae. Now this plant is also used as a biofertilizer. Let us talk about the next field, which is crop biotechnology actually is a very wide field of science, which where microorganisms are utilized. Not only that, the different genetically modified plants are being cultivated to provide better yield of crops. Now, not only in the agricultural field, in the field of animal husbandry, biotechnology also plays a very important role. Generally, the methods used in animal husbandry are artificial insemination and embryo transfer. Now, animal husbandry, actually in lower classes, you must have learnt about the rearing of cattle, okay, and of course, animals which are useful to produce, which can provide meat as well as provide milk and milk products. Now, these cattle, not only cattle, there are other animals like pig and goats, okay, lambs, the sheep, which can be reared. So it is the cattle rearing or the sheep rearing that fall under animal husbandry. It might be even rearing of horses. So the animals, if they are reared, then for human welfare, it comes under animal. It can be studied under animal husbandry. Now, under animal husbandry, there are two main methods, okay, that are used. One is to increase the number of to multiply the number of animals. Two methods are being utilized. One is called as the artificial insemination and the other one is the embryo transfer method. Now, what is this an artificial insemination and embryo transfer for? Actually, it helps to improve the quality as well as the quantity of animal products and animals and also the animal population with more strength, which are developed for even hard work. So let us see how this artificial insemination in cow helps out. Now, artificial insemination is nothing but the process by which semen, that is the sperm containing fluid, is being manually introduced using a catheter and introduced into the reproductive system of the female cattle. Cattle or maybe pig, it might be cow, a buffalo, pig or horses, any kind of animals that are being reared. Now, this is done manually so that the female organism conceives the next generation, conceives the offspring, and easily and in a better way with a better quality next generation offspring. Okay, so not only that, even embryos can be transferred. Now embryos, now how this embryo is being transferred, what exactly is done? Actually, in vitro fertilization process, the IVF process, which is being done even in animals, actually for a better and healthy embryos that could be produced. Of course, not only single but multiple embryos can be produced. These multiple embryos can be introduced artificially into the female organisms, female animals, and that may help in the growth and production and growth of the new offsprings, which are very healthy as well as good quality with desired characteristics. Okay, so embryo transfer also helps a lot in animal husbandry. All these, of course, can be done. Now, I told you about in vitro fertilization, which can be done in the lab conditions, in the control conditions, and the multiple embryos which can be developed in the lab can be introduced into the reproductive system, into the uterus of uterine sacs of the female organisms of animals, various animals, and of course, new offsprings and better quality of offsprings can be produced through biotechnological process. Okay, next field is human health, wherein the diagnosis and treatment are two main aspects of human health management. The biotechnology helps to identify the role of gene, actually, if any, in the disease of a person. So not only that, the diagnosis of various diseases like the heart diseases, the diabetes, AIDS, dengue disease, the COVID-19, etc. All these is possible by biotechnology, and it can, so that it can be treated early, right? Products like hormones, vaccines, antibiotics can also be produced with the help of microbes using biotechnological process. If we talk about hormones, biotechnology is useful for the production of hormones like insulin, somatotropin, thyroxine, and blood clotting factors. Not only that, hormones like insulin, which is used in the treatment of diabetes, can be produced with the help of bacteria now. Earlier, insulin was being collected from pancreas of horses. Of course, it used to be very costly for people who were suffering from diabetes. But now, due to biotechnology, insulin can be prepared by inserting the human insulin gene, okay, into the genome of the experimental bacteria. Like E. coli, I had told you, E. coli, you have agrobacterium kind of bacteria. There are various bacterial strains that are being used as experimental organisms, experimental bacteria. And when these genetically modified bacteria grow and multiply in the lab, in mass cultures, then of course they produce lots and lots of these hormones, the insulin. And nowadays insulin, of course, is available in the next shop, okay, medical shop, medical store, and can be provided to the people under treatment for diabetes. This particular bacteria actually produces the insulin as a byproduct, maybe, that can be extracted and used to treat diabetes. So in this way biotechnology is useful, plays a very important role in the production of hormones under human health. Not only that, vaccines and vaccination can also under vaccination drive are very, very important. You know, nowadays it is very important to have the COVID-19 vaccine, right? Now this vaccine is actually an antigen containing substance, a protein that is given to acquire either permanent or temporary immunity against a specific pathogen or a specific disease. Okay, earlier these vaccines were prepared with the help of partially killed or fully killed pathogens. Now there used to be a risk where the partially killed pathogens may become active and would cause the disease again instead of producing immunity against it. So nowadays, because of biotechnology, due to biotechnological process, safer vaccines can be produced by isolating the gene from the pathogen itself and using it to produce the um, antigenic proteins or antigens in the laboratory. These antigen these proteins that act as antigen can be used to keep the immune system active against the diseases like polio. You have polio, you have vaccine, you have hepatitis vaccine. Nowadays, you have COVID-19 vaccine also that remain active for long duration. Now, there are certain vaccines which come under edible vaccines. Edible vaccines, how can any vaccine can be edible, right? Yes, vaccines are coming as edible vaccines nowadays. For example, transgenic potatoes are being produced, okay, that act against a bacteria called as Vibrio cholerae. Now, this Vibrio cholerae causes cholera disease, okay, whereas Escherichia coli is again another bacterium that may cause certain alimentary canal problems. Now, the transgenic potatoes are being produced by gene manipulation in the potato plants. Consumption of these raw transgenic potatoes generates immunity against cholera and a disease caused due to the E. coli bacteria. But these potatoes, remember, should not be cooked at all, as it may degenerate the biomolecules that are produced and impart the immunity. And so they should be always consumed raw. So here you can see the diagram showing how the transgenic potato can be produced by biotechnological process. Actually, the desired gene is isolated from the human pathogen, okay? That pathogen, that is the disease causing germ or microorganism, and this gene is being incorporated into the DNA of a virus, right, which might be a plant virus, of course, that should infect the potato plant. Okay, these infected pieces of potato leaves are being taken and grown by tissue culture. The whole plant can be developed by the tissue culture technique. And when potatoes are being produced, these potatoes are transgenic potatoes. Okay, so if you consume the raw potatoes, they help in developing immunity against the pathogen. So of course, transgenic crops can also provide immunity when we use biotechnological processes. Now let us talk about another important biomolecule called as interferon. Now, interferon is a small sized protein molecule used in the treatment of viral diseases, many viral diseases. Actually, it is produced in the blood. Okay, nowadays, of course, transgenic E. coli, Escherichia coli, the bacteria are used for the production of this interferon by biotechnological processes, and of course, interferon is being generally given for as an antiviral proteins. Under human health, biotechnology plays a very important role in another therapy called gene therapy. This is being used to treat genetic disorders, which has become possible only because of biotechnology. For example, genetic disease, a disorder called as phenylketonuria. It is a genetic disorder that arises due to certain genetic changes in the hepatocytes. Hepatocytes, of course, are the liver cells, okay, of the liver. Now, these hepatocytes are being treated by gene therapy method, and this method actually is called as somatic cell gene therapy. Somatic refers to the body cells. Of course, the liver cells are body cells, and the genetic changes can be caused under this gene therapy, and the genetic disorder can be treated. Next, let us talk about cloning. Cloning is the production of replica of any cell or organ or an entire organism. Okay. Now, of course, you must have heard about the different types of cloning. Have you heard? There is reproductive cloning and therapeutic cloning. Therapeutic cloning refers to that particular cloning where the production of replica is done for the production for human health, under human health. And reproductive cloning, of course, is used for producing the better quality of springs, right? Now, if you have heard about cloning of the sheep, Dolly sheep, which was actually born in Scotland by cloning technique in 1996. The nucleus from the udder cell of sheep of the Fin Dorset variety of sheep had been introduced into the enucleated. Enucleated refers to removal of nucleus from the ovum, from the egg cell of the Scottish sheep. Now then the ovum was allowed to develop in the uterus of the Scottish sheep itself, and thereby this particular egg which was in which the nucleus of the udder cell was introduced, which was diploid, of course, started acting as zygote cell and started dividing into an embryo, dividing by mitosis, grew into an embryo, and the embryo gave rise, of course, to a normal lamb, a small baby lamb, baby sheep, called as, which was named later as Dolly. Now here it showed the characters, of course, of the Scottish sheep, of course, because the nucleus was from that particular variety of sheep. So cloning was in 1996 such a successful event that slowly cloning started taking up the reproductive arena of animals. Not only that, even therapeutic cloning became very important under biotechnology, and biotechnology plays a very important role in the cloning processes. Let us see what is reproductive cloning. A clone can be produced by fusion of nucleus of somatic cell with the enucleated ovum of a female organism, right? Thus, there is no need of sperm to produce a new organism, remember. So this is the strength of the cloning technique under biotechnology. The therapeutic cloning, under that, the stem cells can be derived from the union of somatic cell nucleus with the enucleated egg cell. Now here various diseases can be treated actually with the help of these stem cells. Similar to these cells, the genes can also be cloned to produce copies of the same gene required for gene therapies, right? And this can be done through microbes. Of course, experimental bacteria can help us out for this process. Controlling the inheritance of hereditary diseases, continuation of generations, enhancing the specific tendency may become possible only due to cloning techniques nowadays. Human cloning is also possible, but it is banned because of various reasons. You must be knowing about it. Next is biotechnological role in industrial product, that comes under white biotechnology, wherein various industrial chemicals can be produced through less expensive processes. For example, alcohol production from sugar molasses with the help of transgenic yeast is a very basic type of biotechnological process which helps in production of industrial products. Not only that, biotechnology plays very important role in environment, and of course microbes are being utilized under these techniques. Environmental related problems are nowadays solved to certain extent due to biotechnological processes like the treatment of sewage and solid waste. Sewage that is released in water bodies causes decrease in the levels of oxygen that affects the aquatic life, isn't it? Because the levels of oxygen, the biological demand of causes biological demand of oxygen in the water body, causing problems to the aquatic life, causing damage of life of aquatic organisms. Using these microbial techniques, sewage can be treated, and then when it is let into the water bodies, it can be, of course, it can lead to less pollution of water bodies. Not only that, the microbes that are used on large scale while production of compost by treatment on solid waste material. bioremediation, biopesticides, biofertilizers, biosensors, or bapre. All these are few new concepts that are being utilized under in biotechnological methods. Bioremediation means either absorption or destruction of toxic chemicals and that are harmful pollutants with the help of microbes. For example, you can see, of course, under biofertilizers, we talked about Anabina species already. Anabina nostoc species that acts as biofertilizers. Not only that, let us talk about bioremediation first. For example, bacteria like Pseudomonas, Alcanovorax, etc. are used for cleaning the hydrocarbon and oil pollutants from the soil and water. You must have heard about the HCV bacteria, the carbonolastic bacteria, right? So these clean up the oil spills on the water bodies. Not only that, even the oils spilled in the soil, it can be cleaned away by these microorganisms. Bacteria like Deinococcus radiodurans is genetically modified to absorb the radiations from radioactive debris. Okay, you can see pictures of Alcanovorax and Pseudomonas that help in removing the pollutants from the water bodies and the soil. Now, next, if the plants are suppose used for absorption of toxic chemicals and harmful pollutants, then that remediation is termed as phytoremediation. Phytos refers to plants. Okay, so plants which act as a remedy towards cleaning the toxic chemicals. Now, so they are called that process is called phytoremediation. For example, the fern Pteris vittata can absorb arsenic from the soil. Not only that, genetically modified Indian mustard can absorb selenium from the soil, which is a heavy metal. Now, the sunflower plant, of course, can absorb uranium, heavy metal, and arsenic, which is a metalloid, of course. It's toxic chemical from the soil. Grasses like alfalfa, clover, and rye are also used under phytoremediation. Now, you can see pictures of Pteris vittata and alfalfa shown there that are used for under phytoremediation. Even biotechnology plays a very important role in food industry. Food like bread, cheese, wine, yogurt, vinegar, etc. are produced with the help of microbes, and of course you have learned all these under a separate chapter called Introduction to Microbiology already. So let us move further. Biotechnology also plays a very important role in DNA fingerprinting. Now, what is this DNA fingerprinting? Let us see in brief. DNA sequence of each person is unique as the fingerprints of that individual. The identity of any person can be established by available DNA, which is simply termed as the process which is DNA fingerprinting. Actually, DNA fingerprinting is generally useful in forensic sciences to identify criminal, and it is also useful to identify the father of a child, any child, if at all demanded or questioned. Now, research is performed nowadays at using DNA fingerprinting at the Center for DNA Fingerprinting and Diagnostics, Hyderabad in India. Right, that is all about the different role and the commercial applications of biotechnology. Students, thank you for joining me in this session. I wish you all a very good life and a good day. Enjoy yourselves. Thank you, and let us continue in the next session, the same chapter. Thank you, all of you. And stop streaming.